haven't been as connected with our department as many familiar faces. It's great to come out today. So it's really a pleasure to introduce Lisa Ballance. Lisa Ballance is a distinguished alumna of our program. She received her PhD in 1993 during the time of work at Cneter in Navy, and is now the Director of Protected Resources Division at Southwest Fisheries Science Center, which is a part of NOAA. She sets research priorities for 7 sciences programs and 80 individuals, and is responsible for the day-to-day management of the division resources. She joined NOAA in 1988 as a graduate student research associate, and began working in her PhD that focused on community and physiological ecology of seabirds associated with yellowfin tuna and spotted civet owls in the eastern and tropical Pacific. Postdoctoral research was at the National Research Council, where she looked at comparative cetacean ecology in the tropical Pacific and tropical Indian Oceans, and she started working, I guess, full-time at the Southwest Fisheries Science Center in 1996 and became a leader of the program in 2001. So her research has involved a strong ecological component and is heavily focused on marine mammals, seabird ecology, and oceanic systems, ecological trends in space and time, and an ecosystem-based approach to management. And I'm really thrilled to have her here today to share with us some of her current work and talk about some good old stories from years past. Thanks for coming, Lisa.
Thank you for that great introduction, and thanks so much for the kind invitation to come speak to you. It's a real thrill to come back. I don't think I've been here since I graduated 20 years ago. I'm going to talk to you a little bit today about some of the recent research that we've been doing in my division, focused on hotspots, specifically cetacean species richness patterns in the Oceanic eastern tropical Pacific. Hotspot, that's a term that has been around for quite some time, and it's probably familiar to most of you. Hotspots were, the concept was first made popular in the late 1980s by Norman Myers, who applied it to terrestrial ecosystems. Since that time, there's been a great embracement of that term and the concept by both terrestrial and marine ecologists. The concept of hotspots is valuable in a conservation biology framework because the idea is that you can locate geographic regions of high species richness, you can protect those regions and get a bigger bang for your conservation buck. The exact definition of species richness hotspots varies. Here's a couple of those definitions. Regions where exceptional concentrations of endemic species are experiencing exceptional loss of habitat, or simply areas that are particularly rich in species, rare species, threatened species, or some combination of those. So again, I'm going to tell you a little bit about our research on hotspots in the Oceanic Pacific, but I'll give you the punchline here. My advisors once told me that if you want your audience to stay with you, tell them the punchline at the beginning of the talk. And here it is. Cetacean hotspots, we don't think they represent what we had expected them to represent. And I'll come to that conclusion at the end of the talk. This is a diverse audience. I think a lot about oceanic ecosystems, but based on the posters that I just saw over the last couple of hours, there's a wide range of disciplines and taxa in this room. So I'm going to give you a guide here by way of 3 introductory pieces to the talk before we get into the hotspot section. And I'm going to really start very broadly and focus down, and I'm going to talk initially about science applications, and then I'll get into the nuts and bolts of science. So I'm going to start with an overview of NOAA and Living Marine Resources. NOAA is a very familiar agency to me, but perhaps not to many of you. I remember when I was in academia, and NOAA was a fairly vague concept. From there, I'm going to narrow down and talk just about one geographic region that NOAA does a lot of research in, the Eastern Tropical Pacific. And then before I talk about cetacean biodiversity hotspots, again, because there's such a breadth of expertise in this room, I'll give you a crash course in cetaceans, what I call Cetaceans 101, and finally talk about cetacean biodiversity hotspots in the Eastern Tropical Pacific. So let's begin with NOAA and living marine resources. Probably all of you are aware that NOAA is a federal government agency. Our claim to fame is predicting the weather, predicting hurricanes, maintaining satellites, but there are a lot of other branches of NOAA, and the one that I'm a part of is referred to as NOAA Fisheries or National Marine Fisheries Service. Our mission, you pull it off the website, is stewardship of living marine resources through science-based conservation and management and the promotion of healthy ecosystems. I'd like to say in a nutshell, our mission is protecting our oceans, keeping them healthy. I underline science-based here because of the branch of the National Marine Fisheries Service that I'm a part of. That is the Science Center branch. There are 6 science centers nationally located in each of the 4 corners of the contiguous U.S., as well as Alaska and Hawaii. And the focus of those science centers is science, conducting research. Each science center is paired with a policy-focused region that deals with policy enforcement management. But the science centers themselves, because they conduct research, they're located on university campuses. And that's no accident. We're located on university campuses because we conduct research. It's our focal mission. The research foci is sustaining fisheries, protecting marine species, and conserving marine habitat, and its applied research is guided by legal mandates. An additional focus of ours is education and outreach. So, for example, many of us that do research in NIMS are also adjunct faculty members. I myself have an adjunct position at Scripps, Institution of Oceanography, where I teach and work classes and advise doctorate students. I'm going to focus obviously on the Southwest Fisheries Science Center, which is located in La Jolla, California. It has research divisions that focus on fish and abalone, Antarctic krill, fish and their predators, environmental data products, and one for which I'm the director of, marine mammal and turtle division, which clearly focuses on research on marine mammals and turtles. The mission of our division, as well as similar divisions across the country, is primarily to assess marine mammals and turtles relative to management objectives in U.S. exclusive economic zone waters or waters where the U.S. has a vested interest. So that's really the cornerstone of what we do. Associated with that, we identify threats and try to mitigate them, educate and build capacity, and overall advance science and management and conservation. This mission stems directly from our 2 primary mandates that will be familiar to all of you, the Marine Mammal Protection Act and the Endangered Species Act. Those acts specifically talk about the ultimate goal of the ecosystems and the animals that live in those ecosystems, and they have elements in them that direct our research. So, for example, both talk about stock structure or distinct population segments. They tell us that we need to understand how populations are structured in order to effectively manage them. They talk about estimating population size and monitoring trends so that we can make sure that particular stocks or populations don't drop to levels that are unrecoverable. I will point out in this little box that our mandates require research outside of U.S. waters. That's because the species that we manage, marine mammals and turtles, do not recognize geopolitical boundaries, and so we have to conduct research outside of U.S. waters. And indeed we do. Our primary geographic responsibilities include the California Current, which is an ecosystem that runs the entire, along the coast of the entire U.S. West Coast, but it extends northward into Canada and southward into Mexico, and seaward about 300 nautical miles offshore. It's a large area, 1.3 million square kilometers. A second major focus of ours, and one that I'll talk a lot more about today, is the Eastern Tropical Pacific, approximately between San Diego, Hawaii, and central Peru. It's a huge area, larger than 20 million square kilometers of open ocean, as well as the exclusive economic zones of 11 non-U.S. nations. And then we also have a primary focus on Central Pacific ecosystems. I've drawn the polygons around the Hawaiian archipelago and the protectorates of Johnston and Palmyra. There are a whole host of others out in the central and farther western Pacific that the United States is responsible for: Pound, Baker, Jarvis, Wake, those atolls such as that. Any one of these is a very large area, 2.2 million square kilometers in the case of either one of those. So these are our primary geographic responsibilities, but in addition, we conduct research around the world. Again, this is because the mammals and turtles that we manage are pan-Pacific, for example, in many cases. So we do a lot of research in the far western Pacific, Southeast Asia. We do a lot of research in Antarctica, some in South America, and even some around Africa. Because our geographic regions of responsibility are so oceanic, our ship time assets are critical. And we've been fortunate historically to have 240 sea days at our disposal for 2 NOAA research vessels, typically in the fall. As the NOAA fleet ages, our ship time is becoming more and more restricted. The David Starr Jordan was retired in 2010, and the MacArthur II is now in warm layup. So we're without research vessels. This is common not only to the NOAA fleet, but also to the UNOS fleet, the university fleet. We're fortunate to get a replacement for the David Starr Jordan, the last one, which will come online this fall. So we use those ships to do 2 types of surveys. One of these is what I refer to as abundance and ecosystem assessment surveys. Those surveys are conducted in those geographic regions that I pointed out to you earlier: the California Current, the Eastern Tropical Pacific, and the Central Pacific. These are really monitoring cruises that allow us to assess and monitor the status of ecosystems and the species that are in them. We like to conduct those on a 3 to 4 year cycle. And then the second, these cetacean and ecosystem assessment surveys use a multidisciplinary approach, and we've used that since 1986. There are 3 main components of these cruises. A big one is abundance. We want to make sure that we can come back with data that will allow us to estimate abundance for all the marine mammals and turtles in that geographic region. A second major focus is biology of those target species. So we collect skin and blubber biopsy samples that allow us to investigate population structure using molecular genetics methods, for example. We use those same biopsy samples to look at reproductive condition by doing hormone assays. We have a very comprehensive passive acoustics program that's focused on cetaceans and their vocalizations. And then finally, a third component of this is ecosystem assessment, where we try to characterize the ecosystem as comprehensively as possible from top to bottom. So we collect physical and biological oceanography, which represents habitat for those target species, low and mid-trophic fishes and invertebrates, which are their prey base, as well as other apex predators, including seabirds, which represent competitors and commensals. So I mentioned there were 2 types of research cruises that we did. The second type is a question-based cruise, or often referred to as process cruises. These are cruises that are conducted on a single time only. They're focused on answering specific questions. Here's a whole list of questions that we have focused our research cruises on a one-time basis. I'll draw a circle around this one. This is a cruise we conducted in 2004. The question was, how many humpback whales are there in the North Pacific? The reason we conducted that cruise is because humpback whales are currently listed as endangered, and that's the case with all of the baleen whales. That's due to commercial whaling that depleted their populations down to very tiny numbers. The gray whale right now is the only one of the large whales that's been taken off the endangered species list. Humpback whales are recovering and have been for quite some time. In order to take them off the endangered species list, we have to know how many are there. And so we conducted this cruise in 2004. That was actually an entire North Pacific cruise. answer to that question. It turns out that humpback whales in the North Pacific have recovered to the point where we are going to work towards having the agency down list the species. We do other types of field work that includes aerial surveys for species that are very coastal or difficult to see from ships. Marine turtles are an example of that. We do shore-based surveys for gray whales, both on the southbound and the northbound migration, because they are so coastal. You almost never see a gray whale from a large research vessel. And then we do small boat-based research to monitor health and condition of cetaceans and pinnipeds in our backyard here in the Southern California bayou. In addition to that, we have 6 research laboratories on site. They focus on molecular genetics, photogrammetry, stable isotopes, hormone assays, life history and condition, and cetacean acoustics. And the division, as Dan mentioned earlier, is 8 programs. 7 of those are science-focused, and 1 is a support program. And we have more or less 80 individuals, including quite a few doctorate students and postdocs as well. I'm going to give you 4 examples of the type of research we do. All of it is applied research, but it rests on the foundations of very solid, basic science. The first one is our extensive cetacean passive acoustics program has allowed us to develop algorithms that allow us to identify cetacean vocalizations to the species level. This was actually a project that was done by a Scripps doctorate student. This is a couple of examples of spectrograms of dolphin whistles, and this student developed various measures of those dolphin whistles, and then developed algorithms that allowed her to classify these calls to species level. We were validating that with the visual observations. The application of this research is that passive acoustics can ultimately, we hope, be used to monitor abundance and trends of some species without using expensive ship time to do that. Second example I want to mention has to do with our extensive gray whale abundance and calf production time series. We found that gray whale calf production tracks ice condition. And so here's a figure that shows you calf production in red, and you can see over time that it fluctuates quite widely. Some years are really high calf production, some years are low calf production. The blue line shows an ice index for the previous year. You can see that those 2 indices track each other quite closely. The application for us is that gray whales clearly are indicators of the ecosystem effects of climate change. Gray whales feed in the Bering Sea, areas where ice condition is quite impacts the prey base there quite well. So this is something that we're monitoring quite closely. A third example of research has to do with critically endangered leatherback turtles in the Pacific Ocean. They breed in the far western Pacific, and we have put satellite tags on some of those leatherback turtles on breeding sites, breeding beaches in the far western Pacific, and tracked them as you can see that these animals migrate all the way across the entire Pacific Ocean to feeding areas right here in the California current. In fact, Monterey Bay is a very important feeding area for leatherback turtles. It's a high-use area, and this satellite tracking has allowed us to identify those high-use areas. The application is that critical habitat for these leatherback turtles can be protected without having to protect the entire ocean. In fact, the leatherback turtle was just recently declared the California State Marine Reptile. And my final example here is our use of ecosystem data that we collect concurrently with species data on our research vessel surveys. We've determined that we can use these ecosystem data to predict species density. Let me show you what I mean by this. Here are our ship tracks on the left-hand side. The blue dots represent sightings of a particular rare species of cetacean, Cuvier's beaked whale, that is known to be particularly sensitive to Navy sonar. The right-hand side is a schematic that depicts the type of physical and biological oceanography that we collect concurrently with the species data, and we've had a whole host of research projects that combine these two data sets into predictive models, so that basically, ultimately, the goal is to look at remotely sensed environmental data and be able to predict density of Cuvier's beaked whale and a whole host of other different species. So here's a density grid that's resulted from one of these models. The application is twofold. One, users of the marine environment, the Navy, for example, in this case, don't deliberately want to go out and conduct their exercises in areas of high species density. So with this predictive grid, they can avoid those high use areas. And second, the Navy and anyone else who wants to go out and conduct any type of exercise or activity in the marine environment that may impact marine mammals, they have to comply with environmental impact regulations and statutes. And this tool allows them to come up with the expected number of individuals that would be impacted by that activity. In fact, we've got an online website where you can draw a user-determined polygon, press a button, and it will tell you the number of individuals of a particular species that are expected to be in that polygon. So there are lots more examples of the research that we do on the website. Now I want to move on to the second portion of my talk and focus down into a specific geographic region, the Eastern Tropical Pacific. Now, when most people think about the Eastern Tropical Pacific, if they've heard about it at all, they think about the tuna dolphin issue. And in fact, that is why NOAA is in the Eastern Tropical Pacific. I'm not sure how familiar all of you are with the issue, so I'm going to give you about 3 or 4 background slides to put us all on the same page. And it starts with this association between yellowfin tuna, spotted and spinner dolphins, and tuna-dependent seabirds. That association forms the basis for one of the world's largest tuna fisheries here in the Eastern Tropical Pacific. In fact, that association is a hallmark of this area of the ocean, the prevalence of these multi-species aggregations. You can find those multi-species aggregations elsewhere in tropical oceans, but nowhere are they near as prevalent as in the Eastern Tropical Pacific. Because the tunas associate with air-breathing mammals, dolphins, and because they attract large speciose flocks of seabirds, they provide a very clear visual signal that allows you to essentially locate surface tuna schools visually. And this is unique in the Eastern Tropical Pacific. Nowhere else would you ever dream about going out on a ship and looking for a tuna school with binoculars. But you can do that here quite clearly, as you can see, by how apparent these surface dolphin schools are. In the 1950s, tuna fishers discovered this association, and they began to use what were known as dolphin sets to catch large schools of very large-bodied tuna. And you can see one of these tuna in this lower right-hand photograph. The dolphin schools were visually located and chased by speedboats into a purse seine net, and the co-schooling tuna followed those dolphins and were also captured. The problem, of course, with encircling the dolphins was that they would drown in the nets, and there was very high incidental mortality of dolphins in the early years of the fishery, so high that 3 stocks that were associated with these tunas were declared depleted under the U.S. Marine Mammal Protection Act. In fact, it's estimated that more than 6 million dolphins have been killed since this fishery began. Here are some abundance trends for 2 of those 3 depleted stocks. The northeastern offshore spotted dolphins have been estimated to be between 19 and 28% of the pre-exploitation levels. Eastern spinner dolphins at 44% of pre-exploitation levels. And then the third depleted stock is coastal spotted dolphins at 42% of pre-exploitation levels. So this tuna porpoise problem, as it was known in those days, was actually so great. It was one of 3 drivers that led to the creation of the U.S. Marine Mammal Protection Act that was enacted in 1972. It was also the primary driver for the creation of the division that I direct, and that was the focus of much of our research for the first few decades of the division. As awareness of the tuna dolphin problem grew through actually the American public, it resulted in revised fishing practices known as a back-down procedure. It was a procedure where the fishers themselves actually got in the water, in the nets with the dolphins, and helped them over the cork line and into freedom, as you're seeing this tuna fisher do with this spotted dolphin. So those revised fishing practices drastically reduced dolphin mortality by about 1990 from literally hundreds of thousands down to hundreds. Here is a schematic that shows the number of dolphins killed on the y-axis over time. On the x-axis, again, you can see that by 1990, the mortality had been reduced to very, very low levels. The expectation subsequent to that was that the depleted dolphin stocks would recover. So our Eastern Tropical Pacific Research Program began in the late 1960s to study this problem. The focus initially was on monitoring the status of those depleted dolphin stocks. Here are some survey track lines of just the Eastern Tropical Pacific, in this case from 1986 through 2006. And the data that were collected on those cruises have allowed us to calculate abundance and track trends. Those data indicate that there is some evidence that those depleted dolphins may be recovering. Here are time trends of abundance in the CVs around those abundance estimates for the 3 depleted dolphin stocks. In fact, especially if you focus on the latter time period, that from 1998 through 2006, you can see that those 3 depleted stocks, the northeastern offshore spotted, eastern spinner, and coastal spotted, have increased at rates of 0.035, 0.092, and 0.077 respectively. So things are looking a little positive. However, if you're a statistician, you'll know that there's substantial uncertainty surrounding the point estimates of abundance. They're very large, and that's primarily due to the fact that the study area is so large and we don't have enough ship time to get those CVs down. If you look at the 95% confidence intervals of the growth rate over the 20-year series, and since 1998, those confidence intervals do include 0, so we can't exclude the possibility that there's been no recovery of the dolphin stocks. Regardless of whether the trend is increasing or not, the dolphins are clearly not recovering as expected, given the fact that the mortality rate has dropped literally to 0 since 1990. And there are 3 main hypotheses that we're currently exploring to explain those abundance trends. Those are these, that chasing encirclement by the purse seine fishery is reducing dolphin reproduction and survival, even if it's not directly killing them. The second hypothesis has to do with an oceanic regime shift, possibly, that has changed the ecosystem to the point that recovery to pre-exploitation numbers cannot occur. And the third hypothesis is simply that our expectation of immediate and rapid recovery is too simple. These hypotheses are not comprehensive and they're not mutually exclusive. I'm not going to talk any more about the research that we've been doing along these lines, but I'd be happy to answer questions about it afterwards if there's time. Meanwhile, even though the dolphin safe label is in place, purse seine sets on dolphin schools do continue, and tuna caught on those dolphin schools are marketed elsewhere under a different label. In fact, the number of sets on dolphins has been relatively stable for the past 20 years at about 10,000 sets per year. So NOAA's Eastern Tropical Pacific program is ongoing, and it represents, without a doubt, the most intensive research effort on marine mammals in the world. That point is made nicely by this paper that came out just last year in PLOS One. It shows the global footprint of the Eastern Tropical Pacific Research Program in terms of number of surveys, number of species surveyed, the proportion of global survey effort, and the number of abundance estimates that have made. So our research has made its mark on the globe, so to speak. And as I said, we continue to study this tuna dolphin problem and the non-recovery and try to disentangle the reasons for it. There have been a lot of other science spin-offs that have come out of this research program. I myself got my PhD out of that research program, and I'm going to tell you a little bit more about some of the other science that has resulted from that. The science on cetacean biodiversity hotspots. But again, before I go too deep into the cetaceans, I want to make sure that you all are with me on what cetaceans are. So here's a crash course, Cetaceans 101. Starting with the basics. Cetaceans are marine mammals. Marine mammals obviously are mammals, and that means they have mammalian characteristics, such as they're endothermic, they are slide young, they're haired, they have distinct physiological and skeletal characteristics. And then the marine part, obviously they obtain food from the sea. Marine mammals are case-selected, they live long and they live slow. Low reproductive rates, long lifespans, and they include this suite of groups. Pinnipeds, which are seals, sea lions, and walrus. Sirenians, which are manatees and dugongs. Sea otters, polar bears. Many argue that fish-eating bats are marine mammals because they're mammals and they obtain their food from the sea. And then finally cetaceans, which include the whales, dolphins, and porpoises. Another very interesting thing about cetaceans is that they're secondary marine forms that evolved from terrestrial ancestors. Here's a cladogram of archaeocetes, ancient cetaceans. All of these are extinct now, but you can see that the ancient ancestor of cetaceans is this family Cetacean over on the lower left, looking for all the world like a terrestrial mammal because it was. From there, we moved to Ambulocetidae, which was a sort of an estuarine form, able to walk around on land, but also quite at home and probably spent most of its time in shallow waters. We get to Remingtonocetidae and Protocetidae, which were progressively families that were more at home in the marine environment, although you can see that they still had forelimbs and were capable of walking around on land. Then we get to the Durodontidae, which is the group, the clade from which modern-day odontocetes and mystocetes were thought to have arisen, and then the final one up there on the right-hand side, Basilosauridae. So it's a fascinating evolutionary story, and there are three major features associated with cetacean evolution along the way. Probably the most dramatic has been reduction and ultimately loss of hind limbs from fully four-limbed organisms that were capable of walking around on land to today's modern large whales. Their pelvic girdle and lower limbs, hind limbs, are greatly reduced to small bones that are encased within the body cavity, and ultimately to the toothed whales that have just two single bones known as the innominate bones in a bottlenose dolphin. The size of those bones, each one is the size of my index finger, and they're buried free floating in the body cavity. So that's a pretty dramatic evolutionary change. A second feature associated with cetacean evolution is hyperphalangy, an increased number of bones in the digits. So here what you can see is the forelimb of a greyhound and the forelimb of a dolphin on the right-hand side. And you can see that the length of the long bones has been greatly reduced. The humerus bone of all cetaceans is completely pulled inside the body cavity, so the elbow joint is at the body surface. The long bones are greatly reduced, and they have increased the number of bones in their digits to deal with the fact that those forelimbs are now functioning to steer, not to support the animal from gravity on land. And then the third major feature associated with cetacean evolution is telescoping of the skull. Telescoping is essentially dramatic changes in the shape and size and placement of the bones of the skull. Here you can see the skull on the top of an archaeocene, one of those ancient extinct whale groups that looks fairly similar to a terrestrial skull of a mammal. A modern dolphin skull is below that, and the different bones are shaded so that you can the changes there. The main driver, evolutionarily speaking, for these changes was the need to get the nostrils up on top of the head for modern-day cetaceans. Modern-day cetaceans fall within the order Artiodactyla. They are completely independent of land. They're essentially hairless, and they have a well-developed blubber layer, as well as a whole bunch of other characteristics that they share. There are two main types of cetaceans, the baleen whales and the toothed whales. The baleen whales comprise the suborder Mysticeti, which literally means mustached whale. There are 4 families and about 14 species currently recognized. Rather than functional teeth, they have these baby plates that they use to string prey. They have 2 external blowholes, which you can see in the upper photograph, and they include the largest animals ever to live on the planet, which is the blue whale. Families in the Mysticetes include the Balinidae, the right whales, the Neobalinidae, the pygmy right whales, Escherichthyidae, the gray whales, and Balaenopteridae, the rorquals. And here is the first family that includes some species that I'm going to be talking about with the biodiversity talk and the part of the talk in the Eastern Tropical Pacific. There are 6 species from this family that we see in the Tropical Pacific. And then the other main group of cetaceans are the toothed whales. They are found in the suborder Odontoceti, which literally means toothed whale. There are 10 families and some 65 species of Odontocetes. It's much more diverse than the Mysticetes. And as the name implies, all of them have functional teeth, although the size and degree of development of the teeth varies greatly depending on the family. There you can see the teeth in the lower jaw of the sperm whale here on the lower photograph. Most of the Odontocetes have well-developed homodont teeth, but some of the families, the males are the only ones that have teeth, and they only have a few. Unlike the Mysticetes, the Odontocetes have 1 external blowhole, and there's 1 extinct species due to anthropogenic factors. The Odontocetes include these families: Physeteridae. This is the sperm whales. This is one of the species that mass strands. And this species is found in the Eastern Tropical Pacific. Kogiidae, 2 species in the Eastern Tropical Pacific: dwarf sperm whale and pygmy sperm whale. Zephyidae, the beaked whales, very bizarre family. 5 species in the Eastern Tropical Pacific. Pygmy beaked whale, that was actually described there during our research. Tropical bottomless whale, formerly known only from skeletal specimens prior to our research. Dense beaked whale, Cuvier's beaked whale, and Bear's beaked whale. The river dolphins used to be considered a single family. Now we recognize that there are 4 distinct families, each with 1 genus and 1 species. This is the group from which we lost one in 2007. It was the Yangtze River dolphin. The Baiji declared extinct in 2007. That animal was seen alive in the wild in 2002. So that extinction represents not only the loss of a species, but a genus and an entire family of mammals. Monodontidae is another family here, 2 species, the narwhal and the beluga. Delphinidae, a very speciose family, 36 species. Some of them are quite striking. This is a southern right whale dolphin, a snaky looking thing without a dorsal fin altogether, which is weird for a dolphin. 15 of these species are found in the Eastern Tropical Pacific. And then the last family, Phocinidae, the porpoises. So there's my crash course on cetaceans. And let me finish with telling you a little bit about what we found about cetacean biodiversity hotspots in the Eastern tropical Pacific. And I'm going to borrow quite a bit from several presentations I've made at past meetings. I want to acknowledge my collaborators on this work, all from the Southwest Fisheries Science Center, Jessica Redford, Paul Fiedler, and Robert Pittman. So I've talked to you already about the Eastern Tropical Pacific. The study area is this blue polygon here. I haven't emphasized two things that are important here. One, it's truly an oceanic geographic area. Oceanic, when I use that term, I mean off the continental shelf. The continental shelf all along the west coast of North America, Central America, and South America is extremely narrow. So you only have to go a few nautical miles offshore and you're really in deep waters, oceanic waters. And then the other thing is that it's large. You know, I've already thrown this number at you, 21 million square kilometers. Gee, what does that mean? It's a big number. Too big to get your head around. So I offer this analogy. This is courtesy of one of my colleagues who's taken that size of the Eastern Tropical Pacific and laid it over top of the North American continent. Another analogy that I like to make is it's an area that's roughly 1/3 larger than the continent of Africa. So imagine that you and a buddy are going to go out and try to determine the abundance and distribution of all of the African mammals. You start at the top, each of you has a bike and a clipboard, and 4 months later you're going to meet down in Cape Town. That's kind of what we're trying to do with 2 ships out here. It's a huge area. Let me say a little bit about the oceanography of the region. There are 3 major surface currents. Those are here. The North Equatorial Current that comes off the California Current and flows westward. South of that is the North Equatorial Countercurrent that flows across the Pacific from the far western Pacific. And then further south of the equator, is the South Equatorial Current that essentially stems off of the Peru Current. So those 3 major surface currents, there are also 2 mesoscale surface features. One is the Costa Rica Dome, that's a cyclonic gyre that basically stems from the North Equatorial Countercurrent when it crashes into the Central American coast. And the second mesoscale surface feature is the equatorial front. It's labeled here just to the west of the Galapagos. It's the region where those 2 large surface currents, the countercurrent and the South Equatorial Current, meet. I'm going to be talking about these areas a little bit more in a few minutes. Temporal variation in this oceanography can be significant, and it's significant on 2 temporal scales. The first is seasonal. So what I'm showing you here are quarterly deviations from the mean sea surface temperature in terms of 4 seasons in these panels. Without getting into the details too much, if you squint your eyes and focus on the colors, you can see that the colors and the patterns are different in those seasons. That's just telling us that there is a fair bit of seasonal variation in terms of surface temperature out there. And then there is also a significant amount of interannual variation. This figure shows you a 100-year time series from 1900 on the left to 2000 on the right in terms of monthly sea surface temperature anomalies for 2 index areas in the Eastern Tropical Pacific. The EU3 index, which is along the equator, and the warm pool, which is a little bit farther north and to the east. But the point here is that you can see those lines are going up and down, indicating that there's a lot of interannual variation out there. In terms of sea surface temperature, you can see the major El Niños of the century, the 82, 83, and the 97, 98 El Niños. These are the seasons and the years, which are data were collected just to give you a feeling for the temporal variability in the study area. And I'm going to come back to that at the end of the talk. So our field research was conducted on these node research vessels that I've spoken about already, and I've showed you these track lines. The data set comes from 10 years of surveys spread over a 23-year time span from 86 through 2006, and comprises a total of 2,116 sea days. The cetacean data were collected using a method known as line transect sampling. It's a fairly standard method of sampling not only animals but plants in terrestrial systems. For us, we had 3 observers on effort on the flying bridge of the ship at any one time, all looking out using these very large mounted binoculars that are 25 power. They allow you to see out to the horizon. And we look for cetaceans from sunup to sundown, weather permitting, when the wind is less than 25 knots. The data set that was collected consists of slightly more than 15,000 sightings from 28 cetacean species. That's about 1/3 of the world's cetaceans. So the analytical methods that we used were occurred in 3 steps. First, we calculated species density. From density, we calculated richness, and from richness, we determined the hotspots. And I'll walk you briefly through each one of these steps before we get to the results. But I will say that our objective in the first analysis was to focus on spatial variation. So we're really looking for spatial hotspots. We controlled for temporal variation by performing these analyses on all 10 years of data as a unit. So we were really ignoring temporal variation. The results are representing a composite, assumed to be synoptic view of spatial patterns. And I'll come back to that assumption at the end of the talk. Calculating density. This is a fairly straightforward, standard thing to do in the world of cetacean biology. The formula is up there, and I'm not really going to go into the details here. I will tell you that we decided that we would use a bin size of 1 survey day, essentially 180 linear kilometers of effort. So for each day, we calculated a density on a species-specific basis. And that gave us a field of 2,116 species-specific density points, 1 for every day of effort. We took that field and then we smoothed those densities using an algorithm. We chose negative exponential distance weighting. We played around with a bunch of different algorithms, and all of the algorithms gave us qualitative results that were pretty robust. I'm just going to show you this one here. We smoothed the densities on grid cell resolution of 100 by 100 square kilometers to get smooth density maps comprised of 2,159 cells. And so these density maps, I'm going to show you quite a few more in the slides to follow. The color pattern is that the gray represents a density of 0, with progressively darker colors indicating higher densities. After we had density, we wanted to calculate richness. We derived richness from species-specific density because if you just look at sightings alone, they're biased. The reason is because if you have an absence of sightings, you don't know if the animal wasn't there or did you just not look there. This is what I mean. Here is a study area and a relatively rare species, Fraser's dolphin. The sightings of Fraser's dolphins are those dots. So you could draw a circle around in this red circle and say, well, there were no Fraser's dolphins there, but is it really no Fraser's dolphins, or did we just not look there? If I add this figure, which gives us effort in terms of the white dots, you can feel a lot more confident that, in fact, there are no Fraser's dolphins inside that red circle because we looked there a lot. So again, we calculated richness based on density because it incorporates effort. So richness was defined simply as the total number of species sighted in a particular grid cell. And again, because we had density, we had to convert density to presence and absence in order to calculate richness. We did this by exploration of a number of different presence thresholds and ultimately came up with 2 rules, one for abundant or widespread species, like the one represented in the upper figure. We considered presence to be the 33% of the grid cells with the highest density. And in the case of rare or localized species, as in the lower map, present was considered considered to be all the grid cells with density greater than 0. So, using those methods, here is our species richness pattern. We didn't have very many of those grid cells with no species cited at all. There were a few of them. And the maximum richness was 19 species per grid cell out of the total species pool of 28 species. So then the final step was to identify our hotspots, and we considered defined hotspots as grid cells containing greater than 40% of the species, or greater than 10 species. And here they are. I've color-coded this now so that anything with less than 11 species is in gray, and the hotspots are in red. And I'm going to spend the last few minutes of my talk talking about this question: what is the significance of these hotspots? So for starters, it's clear that hotspots include important areas, that is, areas of highest density for some species. This is a density plot for Delphinus delphis, the common dolphin. It's an animal that is actually found in great numbers right offshore here in the California current, but also in quite high numbers in the eastern tropical Pacific. The density patterns are shown in blue here, and I've outlined the hotspots in the dark black polygons. So you can see that these hotspots do include many of the areas of highest density for common dolphins. Here's another example of the same pattern for blue whales. In this case, blue whales, areas of highest density are included in many of these hotspots. But when we look at the species in general, the correlation between richness and density was generally low. Here's an example for Papillocephala electra, a melon-headed whale. I've plotted density on the left, species richness on the bottom, and you can see that there's a pretty low correlation between the two, 0.12 in this case. For Papillocephala, the density is highest in intermediate species richness values, and that's pretty typical of most of the species. Here are all 28 species. The mean correlation is 0.26. The range is up to as much as 0.5 for Delphinus delphis, but as low as 0.08 for tropical bottlenose whale and Leucocetus pacificus. And perhaps more tellingly, the proportion of cells where a species was present and encompassed by hotspots was generally low. Here's what I mean. Here's a density plot for Kogia breviceps, the pygmy sperm whale. You can see that it's a temperate species. Its distribution just barely gets into the tropics here. And again, there are those hotspots outlined in dark black. In the case of Kogia breviceps, 40% of the area in which it was present was also encompassed by hotspots. And I'm going to call this the overlap in this and subsequent slides. So that overlap was fairly high for some species. Here's another example, Lagoremus obliquidans, the Pacific white-sided dolphin. We can also find this offshore here in the California current. Overlap high, 58%. But when we started to look at more tropical species, the overlap values dropped considerably. So for Stenobretonensis, the rough-toothed dolphin, for example, the overlap was 18%. Here's the overlap for striped dolphin, 21%. For spotted dolphin, 19%. Spinner dolphin, 11%. There's a couple of the larger dolphinids, Rhesus dolphin, 16%. Pilot whale, 14%. And in fact, the proportion of an area where a species is present and encompassed by hotspots was low in general for all of these 28 species. The mean overlap was 0.2 to 20%, ranging from a high of 58% for Pacific white-sided dolphin to a low of 4% for Fraser's dolphin. So why is this? Why do hotspots encompass such a small proportion of the area in which a species is present? Well, back to oceanography. If you look at these hotspots and you know about oceanography in the eastern tropical Pacific. You can see that the two are clearly correlated. To give you a couple of examples, this hotspot off the coast of Costa Rica is correlated quite closely spatially with the Costa Rica dome, that mesoscale cyclonic gyre that I pointed out at the beginning of this section. You can see that quite clearly with this luscious productivity on this SeaWift satellite image. Here's a second correlation of the hotspot around the Galapagos Islands with the equatorial front, the front between the north equatorial countercurrent and the south equatorial current. In fact, these hotspots seem to be coincident with oceanographic variability. And here's a plot of variability. This is change in sea surface temperature on an ENSO cycle. So looking at the change in sea surface temperature between an El Nino event and a La Nina event. Again, if you just focus on the colors, the darker colors indicate regions where there was greater change. Here are our hotspots. And again, looking at this, we see a lot of correlation with oceanographic variability and the location of those hotspots. We think these hotspots are actually lying along oceanographic edges. So the green hotspot there is the edge between the California current, north equatorial current, and the tropical surface water to the south. The yellow circle is the Costa Rica dome, and the edge of the Costa Rica dome with the surrounding eastern Pacific worm pool. And then the black circle is the edge between the south equatorial current and the north equatorial countercurrent, that equatorial front, which is an edge in and of itself. So now I'm going to take you back to our assumption that, or our point to look at spatial variation and ignore temporal variation. Our hotspots are representing spatial variation averaged through a 23-year time period, and our hypothesis is that the edges represented by these hotspots are shifting spatially over time. We know this is the case. We know that those features do change their location spatially, both on a seasonal basis and an interannual basis. What we also know is that cetaceans out here have strong habitat associations with distinct surface currents and surface water masses. So it's this, we believe, that's causing these areas to be richness hotspots, because they almost always catch the edge of distribution for a great many species, and if you look at a composite of 23 years together, you're ignoring that temporal variation. So the relevant question here with respect to conservation biology really is, are these biodiversity hotspots a valuable way to identify regions of high conservation priority? Typically in conservation biology, what we try to do is identify species-rich areas, and then bingo, that's the area where we want to conserve. But it's really critical to understand the importance of these biodiversity hotspots to the component species. In fact, if those hotspots represent edge habitat, they may be of little value for conserving the component cetacean species here. And understanding the significance of those species richness hotspots to the component species is critical to successful conservation. This is something that we're currently doing further research on with great implications, I think, for the way we think about marine protected areas, certainly in oceanic areas and perhaps in marine systems in general. With that, I will thank you for the opportunity again to speak to you. It's great to come up to UCLA.
Questions? Elise?
Yes.
Has anyone tried to use environmental modeling computer programs to try and see how long it would take these case-selected species to respond to protection?
Okay. How long in time, you mean? Yeah. And maybe you're thinking about the spotted and spinner dolphins, for example?
Yeah.
Not particularly, perhaps with respect to the program that you're talking about. The relevant parameters have to do with reproductive rates, intrinsic rates of growth of the populations. And we don't know very about that for wild cetaceans. Mostly it's been those values come from captive animals. We don't know how relevant captive metrics like that are to wild populations. So it might be simply, as I said earlier, that the populations are not recovering because we haven't given them a long enough time to recover. There's a lot more going on though than I was able to talk about. I mentioned that the tuna purse seiners are continuing to chase the dolphin schools and encircle those schools to catch the tuna below. They let the dolphins out, so the direct mortality is very low. But the chase process itself can go from 20 to 40 minutes, sometimes an hour. So they're actually high-speed chases where the dolphins are being run quite heavily. And we have a lot of research. We've conducted a lot of research that tells us that the reproductive rates of animals in heavily fished areas is a lot lower than it is in areas where those chase and encirclement processes are not quite so intensive. So that's kind of a smoking gun for us right now. Yeah.
So do you do 4-year cycles because you obtain that that's the life cycle of a hotspot?
Yeah. Why do we choose to do our surveys on 3 to 4 year cycles? There are a variety of reasons for that. The big one is that what we want to be able to do is determine trends in abundance. So we really want to be able to detect a decline, for example, if a population is declining. It's great if they're increasing, but we're really worried about the declines. And we've done a lot of modeling work that tells us, based on the geographic size of the area, that if we do a survey at a periodicity of less than 4 years, we lose our ability to detect trends. So that's really the driver behind that. Recently, we started to use Bayesian methods, and they are a lot more robust than frequentist statistics to determine trends. So that might be able to alleviate the need for surveys every 3 to 4 years. But I should tell you that we actually don't conduct our surveys every 3 to 4 years. Scientifically, that's what we say we need to do. The ship time doesn't allow us to do that. Yeah.
So if you were to, I think one of the ideas of having hotspots was to potentially reduce ship strikes. So if you're trying to, or maybe manage fishing in a certain area, so if you're trying to minimize ship strikes, for example, would you still want to avoid the hotspots, or what's the optimal way to, what are the optimal sea routes if you wanted to reduce ship strikes?
Yeah, so actually we've been involved in it. It's funny you bring this up. I know ship strikes on large whales is a huge issue off the port of Los Angeles, and we've been doing a lot of work trying to provide science-based tools to mitigate that problem. And what we've been doing is using our habitat modeling to tell us where we expect different species of whales to occur in greatest densities, and then look at the overlap between the existing shipping lanes with the idea of changing the location of those shipping lanes to reduce the risk. Unfortunately, there are 2 species that are sort of have complementary distributions, humpback whales and fin whales, both of which are ship strike issues here. One is found in the high densities in the northern area, one is found in high densities in the southern. So if you shift the shipping lanes one way, you'll more seriously impact one, and conversely. So it's kind of a tricky problem.
Everyone's going to place. Thank you, Lisa.
Thank you.